Susie,
Rob!
have you ever thought about how you might reconstruct a plesiosaur or what it might look like?
Well, do you know, I don't know that much about plesiosaurs. I'm going to be honest with you. However, I hope that if I was going to reconstruct one, I would do better than famous American paleontologist Edward Drinker Cope, who once put the head on the wrong end, put it on the tail instead of on the neck end.
Let's hope we can do better than that today. Welcome to the Fossil Files. Each episode, Susie and I are going to be discussing a new discovery in paleontology, something that's caught our eye or something that's worthy of discussion, and hopefully we can bring it to your attention too. I mean, there's going to be a lot of diverse topics we're going to get into, Susie, I'm hoping. What are you hoping might come up?
Yeah, loads of stuff. I hope I'm going to learn something because a thing that I find now in my job where I have lots of different things to do. You know, you supervise lots of students, do a bit of research, sometimes do some teaching. I hardly ever get the opportunity to just sit down and actually read some papers that are outside of my field. So I'm actually looking forward to being able to take the opportunity to do that and learn some stuff and hopefully learn some stuff from you as well, Rob.
Completely agree. Less so about what you might learn from me, but I always struggle to keep on top of the new discoveries. It feels like there's, with so many papers coming out all the time, this is a small attempt to try and maybe keep ahead of a little bit of something. So let's start with the paper this week, shall we? So this is Skin, Scales, and Cells in a Jurassic Plesiosaur. Now, I like that title because that is direct, right? That is telling us exactly what is going on. This is by Miguel Marks and friends from Sweden, published in Current Biology this year. So let's start with some context. So, plesiosaurs are giant marine reptiles, the ones that with the long neck and swing paddles. There's lots of them during Jurassic around the same time as dinosaurs, but they are not dinosaurs. I mean, Susie, I don't need to tell you that, but my kids are already annoyed with me, continually telling them out that things that aren't dinosaurs. I go to talks at schools and tell them things aren't dinosaurs. It feels like I'm crushing their dreams.
I know there's loads and loads of things that lived in the Mesozoic, right? Like big reptiles that lived in the Mesozoic. And there's this sort of, I think, kind of public perception that all of them are dinosaurs, or they get grouped under this kind of, heading of dinosaurs as all extinct animals, dinosaurs. In the same way that also even my 12-year-old daughter, who I really would hope knew better, actually said to me the other day that paleontologists are people who study dinosaurs. And I had to correct
Outrageous.
her. I know,
Outrageous.
I
I mean, in your
know.
case, in your case, she's right, but I mean, there are plenty of us out there that don't count for that.
Yeah,
So
exactly.
back on with these plesiosaurs, they often turn up in things like I don't know, all sorts of reconstructions of ancient life. Often lumped in with ichthyosaurs. I was trying to look up in preparation for this what the relationship between ichthyosaurs and plesiosaurs is, and it gave me a bit of a headache. I don't suppose you know. They're just these
Well,
things that live in the sea.
they're most closely related to lizards, aren't they? They're
Yeah,
I think we think that they're probably more closely related to lizards than they are to things like dinosaurs and crocodiles and birds and stuff, the
yeah,
archosaurs. But uh, it's always been a bit of a mystery, hasn't it? Because they sort of appear almost fully formed in the
yeah.
fossil record, and there's not a load of kind of intervening forms. So I think it's people have debated it for a long time, and I'm not really sure that there's a really good answer.
No, one of my hopes is that we can do in maybe 6 months from now, someone will define one of those transitional fossils. That would be an exciting one to do.
That would be really
But
cool, wouldn't it?
no, but for now, we can say that there's been about 200 years of study of these things. So we know they've got long necks, they've got paddles. What they actually look like, though, is a bit of conjecture. I mean, often, I mean, that's kind of common for the fossil record, right? We've got all these bones and shells, but what the actual soft tissues look like is often unknown because we don't preserve them most of the time. So we might be able to imagine the sort of color or appearance of these things, but increasingly we're getting, there's been a sort of flurry of excitement of papers that have looked at these sorts of things. I mean, you've come across those papers, right? Where we've looked at the color of these things and the trying to reconstruct their feathers.
Yeah, it's interesting, isn't it? Because yeah, I used to do tours of the Natural History Museum's dinosaur gallery about 10 years ago to members of the public or longer. And I used to say, oh, we'll never know what colour these dinosaurs are. And then lo and behold,
Yeah,
somebody discovered a way in which we could actually tell what colour they were. But it seems to me that this is very much just based on feathers at the moment, isn't it? I mean,
yeah,
I don't think we have a great I don't think there's it's based on the shape of these structures called melanosomes, which are
hmm
the cells that bear color or bear
Yep.
melanin, which gives certain colors. But I think at the moment that that's only really been demonstrated in feathers, hasn't it, Rob? I'm not sure that it works very well for other types of skin.
I think there are a couple of examples I can think of off the top of my head, but I mean, in this one, we don't really go into that. That's one of the reasons I
No.
was attracted to this paper because I thought, are we going to get some scientifically informed reconstructions of the color of plesiosaurs? But actually, I think this is more about how they function. But I guess before I'm jumping ahead there, let's get into what they actually found. So Marx et al. They report this well-preserved plesiosaur in the Urwelt Museum of Holzmarten.
Have you been to that
So this
museum, Rob? Ever
I
been there?
haven't been to, but have you been?
I haven't been either, no.
It seems like quite a famous one. I was looking it up. They've got lots of good stuff there. It is worthy of a visit, and it's one of the ones where they have. If you are a paleo fan, you might be aware of these logs with loads of crinoids on it, like these giant, massive
Ah, yeah.
floating logs with crinoids on. One of them is there, but they've also got loads of plesiosaurs there. So this specimen has actually been collected ages ago. But these guys, so it was excavated in 1940. Preparation in 2020 uncovered soft tissue traces around the tail, is what they report. Now, one of the things that frustrates me about this is they haven't given this taxon a name. I think they've just called it specimen MH7. I think it needs a name. Can we give it a name for the purposes of this?
Should we
Nigel?
call it not with Nigel? Nigel is a strong name for a plesiosaur. Let's call him Nige.
Nigel. Okay, so we're going to call this specimen Nigel because it doesn't seem to have a taxonomic assignment, which I find a bit weird because it's got a lot of detail in it. But
Does it have a
anyway,
head?
it has a head. I mean, so I don't really know why. Anyway, let's not go into that. They've called it MH7. Being cautious is perhaps fair. And that's not really what this paper is about. This paper is about the bits and pieces around it. Let's have a look. What did they actually do? So they found this specimen and they applied a dizzying array of fancy three-letter acronym techniques to it, including TLM, SEM, EDX. I'm not going to tell you what all of these things are right now, but the point is they're using lots of techniques to get a molecular handle on what's actually going on inside this tissue. So they found fossilized soft tissues along MH7. Sorry, they found fossilized soft tissues of Nigel and are exposed along the dorsal and ventral sides of the vertebral column.
So
Um,
top and bottom
Top
of
and
the
bottom.
tail.
They seem to end up focusing on two particular patches, one on the tail and one on a flipper. So, based on the presence of a distinct internal layering that morphologically corresponds to the stratum corneum, I'm not going to go through all these different layers. They have layers of what they're interpreting as layers on top of this thing. So, they've got all these cross sections of these soft tissues with nice layers and appearing to have subcellular detail in it. So, that's quite cool, right? We don't normally get that kind of material preserved. This is where they've had to do all these fancy histological techniques to try and have a look at what's going on inside and demineralizing some of the tissue. I tried to look up how they demineralized it, but basically they're sort of etching away the bone to look at what's left behind and trying to analyze the tissue of it. Let's have a look then. In the sections of the skin, they find these small bodies that they interpret as fossil keratinocytes. So I had to look up what a keratinocyte was, embarrassingly. It
I had
is
to look it
a
up as well. I had to look it
Okay,
up
that's
too.
fine.
Yeah,
That's
yeah, no.
fine. It's a type of cell in your skin. That's as far as I can tell that might generate keratin, which is an important component of all your horny bits and pieces. But I mean, we'll have to come back to this as to why they would interpret it as keratinocytes, because as far as I can tell, what they've found is small ball type structures or cells. Susie, can you describe to us what we can see in figure
Well,
number two?
so that figure number two, they've got some images of some of these chemical techniques that they've used, and
Mhm.
so they've tried to map some different elements across these different layers that they found, I think, haven't they? And the way
Yeah.
they often show this is they show it with a color map, so different elements like silicon and phosphorus and sulfur and that have are given different colors in these maps. And I don't know about you, Rob, but I think from what I can see from these figures is they've got some layering and they've got lots and lots of phosphorus
Hmm.
and some calcium, which is
Yeah.
calcium phosphate. Now that's bone, isn't it? I mean, calcium phosphate
Yeah.
and bone is like yeah. So would we expect to
Yeah.
see that in skin?
I mean, well, we don't, they don't talk about that a lot, but the calcium and the phosphate is confirmed. I don't think that's very controversial. And you would indeed expect calcium and phosphate in a bone, but you might also expect it in preserved skin. If it was preserved in a certain kind of way, you might get phosphatization taking place. So there might be some bacteria that are replacing the soft tissues with this. So to actually get a hand, I mean, I don't want to talk about this too much because I'm actually writing a paper on this very topic at the moment, and I could end up giving you too much details about trying to discuss whether or not it's phosphate or not, but the or apatite. But anyway,
Yeah.
that's dry chemistry. Let's not get into that. Yeah, so that's what's going on in figure number two. I should have also asked you about figure number one. If we have a look at figure 1D, this is where they've got some of these cross sections of the skin. So what we've got in here is these layers, but also within it are these like balls with tiny bits inside it, which they're interpreting as nuclei. Now,
Yeah.
I find that's a bit difficult. I mean, for me, I mean, on a superficial level, sure, why not? Let's say they're cells with nuclei in, but because I've worked on some of these things from the Cambrian and the Equine, if you want to describe something as a fossilized nucleus, people have written pages and pages of papers about whether or not, you can justify interpreting something as a nucleus. But these guys are just like, yeah, that's a nucleus.
I mean,
Maybe
I
that's
suppose it
fair.
doesn't. Does it matter? I mean, I suppose the thing is that if you've
No.
got some layered material
Yeah.
around the outside of, I mean, because the specimen is beautifully articulated, isn't it? All the
Yeah.
bones are in the right place. They've got a lovely
Yeah.
picture of it, and all its bits are. I mean, it looks like it just died yesterday, and maybe it's been a bit
Yeah.
flattened, but that's
Yes.
it. So if
Yeah.
you've got kind of layered stuff
Yeah.
around the outside of your bones. I like it's sort of the most simple solution is that that is in fact skin, isn't it? And that
Yeah.
then if you have ball-like structures within something that you think is skin, looking similar to what you might expect cells to look like, do you not think that's kind of a
No,
reasonable
I think you're right. I
option?
was being a bit unfair because I'm thinking of these things from the Cambrian and the Equine, where whether or not it has a nucleus is a massive important difference, or whether it is a
Okay.
eukaryote or not. In this case, yeah, fine, why not? Let's go with it. It's got some nice layers. It's got some subcellular type preservation. Yeah, let's say that. That's fair.
I think it's fair because,
Yeah,
I mean, what else are they?
but I don't know. I don't
Could they
know.
be bacteria, Rob? Could they be bacteria?
Well, we're going to come back to that when we get onto the melanosomes, I guess. So they've got these keratinocytes that have become flattened. So they've got this idea that, and they compare it to a reptile skin in E and F. So there's the fossil one, which has got these sort of cells and balls, and then they compare it with a reptile skin. And yeah, I can see that. They sort of change shape as they get closer to the surface, which is apparently something that skin keratinocytes might do. So yeah, fair enough. But yeah, let's get back to your specific question though, bacteria and melanosomes, because the other thing they're looking at is melanosomes. So, as you already told us, melanosomes are these subcellular structures that hold melanin, which is this important pigment. So, the actual shape and distribution of these tiny little subcellular things is really important for helping us reconstruct fossil color. Now, these guys aren't going that far on reconstructing fossil color, but they are interpreting it as melanin and that are melanosomes, which is really important. So, that's what's going on in that figure you described with these tiny little sausages.
Yeah.
I mean, that figure, I mean, I've tried to visualize melanosomes under the ASEM, under the scanning electron microscope myself, and I didn't have the touch. It was really hard. You had to be quite the master on the instrument. And then Sarah Gabbett, who I was collaborating with on, came in and did it, and she was like, yeah, there it is. I was like, okay, right, I don't have the touch in order to do that. So that's technically quite difficult to get a hold of the images. Well, having said that, I couldn't do it. That doesn't necessarily mean it's technically difficult. But they have these nice little sausages that are the right size and shape. But as you pointed out, bacteria, some people have spent a long time. I mean, in the early melanosome papers, a lot of people spent a lot of time arguing about whether things were bacteria or melanosomes because they're roughly the same sort of size and shape. So I guess that is a bit tricky, but that's why you might end up using some of these analytical techniques.
I think the it's a little bit less controversial that melanosomes preserve now, isn't it? At least in
Oh, yeah,
feathers
yeah,
it
yeah,
is.
yeah, yes,
And
absolutely.
one thing that I'm thinking though is just looking at the picture there, we've got part F, and that's a sort of zoom out, and then they've zoomed in on this
Yeah.
patch of melanosomes in G. But in and you can see them in F. You can see they look like a whole load of kind of small short worms lying in a small
Hmm.
patch,
Yeah.
but you can't see them anywhere else. So these melanosomes, they appear to be relatively restricted.
Oh,
Um,
yeah, yeah, yeah.
with
They
the
are.
area where they're preserved.
Yeah, that's right. You're right. And indeed, that they say that themselves. They talk about them being in concentrated little bundles, which I think is probably reflective of reality, right? That is what if you take a histological tissue from a modern organism, you're going to see them concentrated in little bundles. So I think that's fine.
So when you do that, does that because melanosomes hold melanin, which gives pigment, does that mean is them coming together into a bundle? Is that something that happens after during fossilization, or
Oh,
as a result of preservation, or was it spotty? Are we
good
seeing
question. I actually don't know how to answer that. There's a whole team of people in Cork that are busy doing these things repeatedly,
mm,
looking at modern tissues, fossil tissues, how these things get transformed. Uh, That's a good question. We should ask them. Maybe one day we'll get guests on to do this thing. I don't know. If it
We
depends
could.
if anyone's interested. But I think that's an interesting question, which I don't know the answer to. So we've got these things that they're interpreting as melanosomes. Oh, so go back to your question actually about those debates about bacteria versus melanosomes. One of the things that answered that question was looking at their actual chemistry and
Hmm.
composition. So you can do this thing called TOF time of flight, which is this particular technique that I'm just worrying. Should I try and describe it? No, because I would be embarrassing if I get it wrong. But it's the idea that we're blasting this molecular structure, having a look at what comes off. That's so trying to characterize it. It's a way of looking at, does it have the same molecular characteristics of certain kinds of biomolecules that we might anticipate today? So that was one of the things that was confirming these things is actually melanin, is that they look like melanin in this sense with this particular technique. So these guys apply that here, but they don't actually end up confirming it as melanin. They say that it looks like, I'm gonna have to now look up the phrase.
They seem to find,
Asphaltene. Right, yes. So they say that these things are like asphaltene. Yes, sorry.
so they find all these sort of organic compounds, don't they? Sort of
Yeah.
carbon-based compounds. So I presume that asphaltene is like asphalt, is it?
Yeah, so
So
I had to look this up. Basically, it's just organic mush.
yeah, yeah,
I
yeah, yeah.
imagine if you took a whole load of organic stuff and cooked it, you'd end up with this, these, what do they describe it as? Polycyclic aromatic hydrocarbons, or
There you go.
PAHs. So what's happened is basically we've got cooked organic matter. It
Which
would
I
be
mean is not
nice.
it's not that surprising, is it? Because
No,
how old is
no.
this? This is early Jurassic. So what is it?
Yeah.
180 million years, something like that
Yes.
older?
Yes. Yes. Yeah.
It's been dead for a long time.
It's been
We
dead for a
might
long time.
we might not expect it to be perfectly, to have
No,
perfect
no,
perfect biomolecules and all its bits still intact, mightn't we?
indeed. But I mean, you can, people have found melanin and other organic things from toxins and other kinds of very complicated three-letter acronyms. So you can get these sorts of signals, but I guess you do need a fossil that hasn't been too much cooked. And I guess that's what's happened here. Presumably it's been subjected to some high temperature, high pressure during its history, and it's just left with this sort of organic kind of mess. So in some senses, the analysis didn't really confirm what they were hoping to, but hey, they reported it. Oh, you should absolutely report null results, right? That's fine. That's good.
Yeah,
They tried to,
definitely.
and
Mm.
if I was a reviewer, I might have said, yeah, well, why don't you do some TOF sims? So and they did. So fine. Great. They've gone some analysis. Oh, I missed out the scales bit. So yes, we've got all this talk of keratinocytes and other things, but they found something else, Susie. Do you want to find out what else they found?
Yeah, I would like to find out what else did they find, Rob?
So this is from the tail. They found these layers, but in the flipper, is the flipper the right word? I don't
Yeah, it
know.
is. I think Flipper's all good.
Okay. So the skin from the right forelimb, this is quoting them now, incorporates small, irregularly sub-triangular structures that we recognize as scales. I quite like that statement because that's very hedging their bet, so that's fine. They
You can
recognize it as
get
scales.
It's funny, isn't it, when you're writing papers and things that reviewers, there's always, when I read papers, I always think, I bet the reviewers have made them put in something there to like caveat their
Yeah,
certainty. So, they
yeah,
say, oh, we're not very happy that these are definitely scales. So if you put in a phrase like we recognize as scales, then it's sort
yeah.
of, it allows some flexibility around their interpretation, doesn't it?
I agree, but I also think they're maybe just being cautious. I think that's fair. So they clearly differ. So this is again quoting them. They clearly differ from the soft, scaleless skin covering the tail, and instead compare closely with the carapace scutes of fossil and living turtles, which is kind of fun. So if you think about what a turtle looks like, if you think about a turtle flippers, they actually have these sort of harder bits, which are scutes, right? So they're not, which compares to say like we're going to come to dolphins later, but dolphins are covered in these soft, squishy skin, whereas reptiles, including turtles, have these like nice chunky bits in them, which they're interpreting as scutes and scales. So they're basically saying it looks a bit like modern reptiles in a sense. They've got these scales. They've got these melanosomes. I mean, they did say that the melanosomes are a little bit different.
From what? Different from what?
So I think I can't remember the specific details, but apparently the distribution of melanosomes is a bit different from what you might see from modern reptiles. But the scales and the layers and these stratum spinosum and stratum corneum, they all interpret that as being roughly comparable to living sea turtles, which I think is pretty cool.
It
Um,
is. Yeah.
yeah.
And
So
perhaps not that unexpected, considering, that they're both living in the sea with
yeah,
flippers
yeah,
and are reptiles.
yeah, that makes sense. I think. I think one of the reasons it gets a bit difficult is you have to think about what are the things that you might be comparing it to. Turtles makes a lot of sense to compare it to. They also talked about snakes at one point in terms of taphonomy. Why do we get this? Maybe the skin was very thin, like it is in snakes, and that might mean it has low preservation potential. Um, because these are all from the Pisedona shale or the Pised. Well, I was gonna. Oh, I forgot at the beginning we were talking about its German name, which sounds super cool. Poseidonei Schleifer formation. So I'm probably going to embarrass myself with that. So we can edit out that me saying these words in German. We don't need to do that. So it's from this Poseidon shale, presumably named after Poseidon. So they're saying that the low preservation of the skin in the Poseidon shale could be a consequence of infrequent burial in low oxygen environments.
So I was a bit so Robert, yeah, I didn't really understand this bit of the paper. And you work on this sort of taphonomy of soft tissues and things. So maybe you can explain this. I the issue that what we have in the Posidona shales are what I thought were relatively low oxygen environments or something that allowed these animals to be there's lots of articulated animals from them, lots of articulated marine
Yeah,
reptiles.
yeah.
So I thought that that would indicate that there was a low oxygen bottom environment, probably quite a squidgy bottom of the
Hmm.
sea, so that
Hmm.
things were sinking into a squidgy bottom and then being
Yeah,
buried very quickly and not being scavenged and eaten because
yeah.
of this low oxygen in the surroundings. Would we not expect that environment to actually preserve soft tissues quite frequently?
Yeah, well, I mean, there has to be a whole recipe of things to make sure that you have the right conditions to preserve soft tissue. I mean, one of them is often seen as low oxygen, which is, as you say, not only just about the keeping away predators, but also a certain area is the idea is that it will reduce bacterial action, but also there's lots of bacteria that work in low oxygen environments. And actually, there's a lot of evidence from some of these experiments looking at decay and decomposition is that if you decay something, it quickly becomes a low oxygen environment anyway. So I guess what their suggestion is that they're saying that why don't you find all these things repeatedly in plesiosaurs is because maybe you don't have many of them preserved in in the low oxygen environments, other than this particular site. I think that's why Poseidon shales or Poseidon or Schneifers or whatever it is might be better for this.
Okay.
But they also raise another interesting hypothesis for why we don't have more evidence of this, is because that maybe the skin remnants were unintentionally removed during mechanical chemical preparation, which I think is probably highly likely. If you've got
I
this
think it's
nice,
very likely.
I
Yeah.
think it's very likely. If you've got this fancy looking plesiosaur and then you see some sort of, it's all articulated, you've got these nice flippers and this nice long tail, and then you see some sort of scraps of something around it, you get out your chipper, you knock it off, you wouldn't think much about it at all.
Yeah, absolutely. And especially considering that this is historically collected, right? So, I mean, it was collected something like
Yeah,
80 years ago, wasn't it? So,
absolutely.
80 years ago, we didn't really have that view of either probably the significance of soft tissues, the fact that it might be preserved in the first place, but also that it might be really useful and interesting, probably wasn't realized at all by the people who were preparing this material.
Absolutely.
But also in the field, I mean, you do field work as well, Rob, right? We both do a bit of field work and
Sometimes.
sometimes when you find things which
Yeah,
only occurs sometimes as well, the way that you find it is you find the bones, isn't it? You
yeah,
don't see the skin first. so
absolutely.
so also in the field there's probably the chance that you're actually preparing things off whilst excavating things as well.
Yeah. As soon as I saw that, I thought that a second explanation was probably far more likely that these were getting chipped off. This reminds me of the go-go. So I work on fossil fish on occasion, and this go-go Devonian thing in Australia. They've got these absolutely beautiful nodules with fish skulls preserved inside them, and they were always acid prepping them out for decades, right? So they take them out, they put them in this low pH solution, and they just leave them there because the pH gets rid of the rock and leaves this lovely, lovely bones. Great. You end up with these pristine looking fossils. But then I think I can't remember who made the discovery. It's going to be embarrassing. I'm sorry for whoever it was. They actually found that there were soft tissues being preserved as well, but those soft tissues were getting completely cooked away by the
Mm.
acid. So they had to change the way they were preparing it. And that's probably applying here because they talk about chemical preparation. Just melting away the rock is great, but if you're well, melting's the wrong word, but if you might be losing the soft tissues as well while you're doing it.
Yeah, and there's kind of increasing evidence that some of these biomarkers, the breakdown products of soft tissues, could tell us really interesting and important things about the biology of animals that
Yeah.
bones can't tell us, right? There's been some recent work on biomolecules and things that could indicate metabolic rates and stuff like that, and various types of physiology and things. So I think that's it's gonna be increasingly recognized, is it? And people
Oh,
are going to be looking for
sure.
these more and more.
I mean, it's difficult because it's a lot more work, right? If I want to find a nice bone and describe the shape of that bone and put it in a taxon, lovely. If I want to do this soft tissue stuff, as evidenced by these guys, right? They've got to go through this demineralization, the scanning electron microscopes, all these extra steps. It's going to slow people down a lot. But well,
That's true, but also paleo has always been a very multidisciplinary topic,
oh,
hasn't it?
oh, sure.
We've always been well, you and I think both come from a geology background, and we basically do anatomy, right? So
Yeah,
we're joining up, biology and zoology and biology, and that's even, at the very start of paleontology, people were doing that,
yeah,
and now really we're just bringing in geochemistry and all these other different techniques that
absolutely,
and actually
absolutely.
organic chemistry, right? Actually being brought in, and so these sort of teams and equipment and facilities that you need to do paleo are just ever becoming ever more multidisciplinary.
I mean, I completely agree with you, apart from your horrible slander of calling me a geologist. No, I'm a biologist by training. So
I did not know that. I thought
that's
you were
okay.
a
That's
geologist.
fine. That's fine. I
I
mean,
like you
in some
less.
senses, that means I've been able to blag my geology credentials enough that people might believe me. Okay, so let's get back to let's get back to Nigel, shall we? Let's have a look. So we've got this idea that Nigel's got this lovely cells inside it. He's got melanosomes and we've got scales. So we're beginning to get a picture of this. And why this is important, I guess, is that we should always take a step back and think about why we should care about this. Is because we haven't had a lot of information about what plesiosaurs might have looked like and what their skin was. And that's important because it's about how they function. So I guess if we now sort of turn the page and move on to the next bit, which is thinking about the interpretation of this. So we now know what they've done. What does this interpretation mean? They talk about hydrodynamic function and thinking about how this thing might have moved around in the water, which I guess not really something that was new from this. I guess we must have these things were living in water and moving in the water. But I guess one of the other things that's interesting about what Nigel has is it's got this. Their interpretation is that the flippers might have had a protective covering. So if it's like a sea turtle, they've got these scales on the outside. Maybe this was important for grazing, and this is consistent with plesiosaur bottom walking and feeding traces, which is their interpretation. Now, this was kind of interesting to me. I didn't know this was going on. So they talk about stomach contents. Well, I was aware of that, but they also talk about this idea that plesiosaurs were walking along the bottom of the sea and grazing. I wasn't aware of that interpretation. Is this something that was used to you too, or is this?
Yeah, it was totally news to me, but I don't know that it was necessarily news to people who know more about plesiosaurs than you and I, because
No.
they do say that this, this idea of like scooping up sediment and us
Yeah.
kind of sifting out all of the things that are living in the sediment, like all the shellfish and
Yeah.
the bivalves and the gastropods and stuff, is actually, they do find stomach contents with that kind of thing in, and that made me think, well, what is the impact of this discovery then? Because if we already knew that they were doing
Mhm.
that,
Mhm.
is this just largely confirmatory? Is it
In some
is it a
sense, I
step
think what,
forward?
yeah, I think it's a step forward in some senses because we didn't know. And I think one of the important differences is the extra piece of information is that they're different from ichthyosaurs. So I think we can say on the basis of this, I mean, I'm not an expert here on this particular area, but it seems to me that ichthyosaurs, these more dolphiny looking ones that everyone's already, very famous ones, they have much more sort of, well, dolphiny like skin. No, I shouldn't say dolphin like because they're not mammals. They are their own reptile thing. But ichthyosaurs have this very smooth, sleek skin, whereas these plesiosaurs have scales and melanosomes, which means that they might have been doing this other stuff, like this walking about and sort of. I mean, I'm still finding it hard to imagine that you'd have this massive long neck just so you could walk along the bottom of the sea. This doesn't sound right to me. But
Well, do you
I looked.
think? I mean, because what was the neck? Because the neck is it's a bit controversial why they had this massive long neck, right? And at least, I mean, some people, or at least I don't know whether this is whether I've just seen drawings of this. It's possible that I've just seen drawings of this, and actually nobody's actually said it in the literature. But the idea that they were just kind of hanging out and then using their long neck to go after things without actually having to move that much. So were they just propping themselves up on the bottom? and then using their long neck to graze over a large area or to grab things from a large area of the water column, a bit like we did. That's what we think with sauropod dinosaurs, right? With the long neck, long tailed
Yeah.
dinosaurs, is that because their metabolism, how did they support this huge body size? And some of the plesiosaurs were big too, right? As well. So
Yeah.
how do they
Oh,
support
some
this
of these things
huge?
are pretty big. Yeah, yeah.
Yeah. So if you have this long neck, you can kind of cover this quite large feeding envelope without really having to move. So could they just have been, basically propping themselves up on the bottom and then like hoovering up everything around them in this big area?
Well, that's one of the things they talk about here with this grazing idea is that when you
Mm,
look inside the stomach, you see all this sediment and gastropods.
yeah.
So maybe they were just, lazy sea cows just sat on the bottom, scooping all this stuff up. But I mean, we also have this idea of them being active predators of, cephalopods and fish. I mean, I don't think you could just hoover up a fish and an ammonite. It's just going to be swimming past. Well, I don't know, but I get this led me to look up some of these feeding traces because I was like, oh, this is interesting. This idea of walking traces. So when I looked up some of these papers, some from Italy and Germany and Switzerland, and well, I don't want this podcast to devolve into being rude, but I'm sure it will do at some point. But I wasn't massively convinced. I see these sorts of like holes in the ground in a line. They're not paddle shaped. They're just blob, 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 blob. And that left me a little bit unconvinced. But having said that, maybe I should be less of a if I have all I've done is looked at the pictures of these papers. If I want to be skeptical, I need to actually read the damn thing.
Well, okay, Rob, now that you have admitted that you're a biologist to me, I'm going to be addressing all my questions about living animals to you because I my usual response is I don't know anything because I'm a geologist.
Yeah,
So, are there animals? I mean, aside from turtles, what do turtles do this today? Because I'm just thinking if they're using turtles as a kind of analogy where they have these scaly limbs, and the reason that they have the scales on the limbs of this plesiosaur is so that they don't basically graze their skin, right? When they're
yeah,
propping themselves up on the bottom. That's the
yeah,
interpretation because the rest of the body hasn't got the scales, so just the limbs.
yes,
So, and they use turtles as an analogy for having scaly flippers. So,
yeah.
do turtles do this?
Yes, some
Oh, they
turtles
do.
do. Some veggie turtles will sit on the bottom and just graze or browse. I can't remember the difference. Great. They're just eating algae and they can sit on the bottom and they can scoop along. But I mean, I don't see a lot of turtles walking. I guess that's the thing that I'm finding a bit difficult about this is the
Oh, okay.
walking bit.
It's
The
the
idea that
walking.
you could sit on the bottom and just graze or browse. Yeah, I get that. I can see that. We have modern reptiles that will do that. But the walking along the bottom, it seems highly inefficient to me and not very likely. But
Well, do you know, there's an interesting thought there as well about how they actually could move their limbs, isn't there? Because there's been quite a lot of work on how plesiosaurs actually, coordinated their flippers to swim.
hmm.
And Luke Muscat at Imperial has built a model, hasn't he? A
Oh, it's super
robotic
cool. They've got a
model.
little robot. It's fun. I've
Yeah.
seen them talking about that video. It's fun watching that thing flip about. It's cute.
Yeah. So, but I think that the general conclusion is that they're almost flying, aren't they? And they're using their flippers
Yeah.
together. I'm actually
Yes.
doing a mime here. I know you can't see me on the podcast, but it is actually really hard to talk about locomotion without doing a mime. Yeah,
Yeah.
it really is.
I'm moving my arms like I'm a plesiosaur now. Yeah.
But if you're sort of flapping your arms in and out, as it were, or up
Yeah.
and down in a
Yeah.
dorsoventral movement, if you're going to walk, you're going to have to move them forward and backwards, aren't you?
Yeah.
So and also kind of sequentially, like not at the same time, unless they're doing a kind of funny sort of hop. So it sort of would suggest that maybe I don't know. Do we have I don't know. Do living animals? Do we see them doing one thing with their arms in one sort of mode and then another? Do you know what I mean? Like it just seems
I think
weird.
that, yeah, I think that's a fair question. I think after this, I'm going to immediately start looking at YouTube clips of turtles in the bottom of the sea, seeing what they do. Um, but anyway, I guess that's the model they're invoking there. So I guess that leads us to the final conclusions then, I guess. So because that we'll reach the end of what they've had to say about it all. So, in summary, this is now quoting them. Our comprehensive morphology, I mean, they've had to slip in the word comprehensive here, but I'm guilty of this. I've done that. I've definitely done that, or
Well,
innovative, or whatever.
they wanted to get it into current biology. You've got to pop in
Yeah,
comprehensive.
yeah. Okay, so I'll put in us in summary, our comprehensive. I can't laugh without saying comprehensive now. That's unfair because I'm guilty of this. Right. In summary, our comprehensive morphological, microscopic, and spectroscopic investigation of the soft tissues of Nigel suggests that plesiosaurs retain reptilian scaly skin throughout the land to sea transition and later specialization for open ocean. So this contrast, and I'm not going to continue to quote them, but they contrast this with ichthyosaurs and crocodilomorphs, Metriorhynchids, etc. So I think that's why the interesting part of this paper lies, right? Is that they're implying that there's something interesting and different about plesiosaurs that we didn't know before. I think that's fair. I think it was worth reading.
Yeah, I enjoyed it. I thought it was an interesting paper. No, I was going to say I feel like I've come away from it knowing more than I did before, which is not always
Good.
how I feel when I read papers, to be
No.
honest.
I guess I often end up thinking about so what at the end of reading a paper, and I think we can say there is a so what here in the sense that there's some new information about how plesiosaurs moving about. I mean, obviously there's still lots of discoveries yet to be made there. These these moving about traces would help, and now that maybe the other way to look at it is that now that they've confirmed that there are these soft tissues preserved and they may have been overlooked, that maybe other people will start to look at their own plesiosaurs again and go, oh, do you know what? That little bit of scrappiness at the tail I wasn't interested in before, or that little tiny bit of stuff that I flicked away with my brush when I was preparing it. Maybe that's worth another look.
Yeah, that's true. That's true. Yeah, I agree.
Okay, well, thanks, Susie. I hope you enjoyed plesiosaur chat. Let's discuss something else next time. Have you got any suggestions, or shall we have a little think about that offline?
I think we need to have a little bit of a think about that because, as I said, I never read any papers, so need to go away and read some.
I think we maybe should take suggestions from our listeners if we ever have any out there somewhere. Maybe we should invite suggestions that would make our lives easier of papers that people want us to talk about.
Yeah, we might have to put some caveats on that because if it's got loads and loads and loads of maths in it, it's just if it's like really hardcore, I don't know, genomics or something, I'm out.
Oh, I was interested.
Out.
When you said there may be some caveats on that, my first thought wasn't about loads of maths. My first thought was, is it going to be about this isolated tooth of an Allosaurus means that this is the most important thing to ever have happened? But we'll have to.
I'm happy to talk about those. Perhaps not in a positive way. That's okay.
We probably should stop whittering. Hopefully, see you next time.
Bye.
Bye, Susie.
Bye, Rob.
The Fossil Files was presented by Robert Sansom and Susanna Maidment. You can find more information, including links to the original papers that were discussed, at fossils.libsyn.com, and you can contact us at fossilfiles.pod@gmail.com.